All right. Is this the clicker? Oh, that's the wrong one. Oh, here we go. Got it. I think I got the right one. All right. Hey, everybody. I'm Dylan Gordon with CFD Research Corporation, and I'm with the in vitro and in silico modeling group there. And it's honestly a pleasure to be speaking in front of everybody to present our data today. I'm here to present, the work that we've been doing for Tissue Chips 2.0, and we are developing a uterine model to assess the effects of aging and reproductive health and disease in women, especially considering microgravity effects on these things. But before starting, I'd like to give a big thanks to our collaborator. If we didn't have their help, we really wouldn't be able to pull this off, so a big thanks to them. Thank you for the opportunity to present our work. I'm going to give a quick overview of what CFD research is capable of. I know we're running a little behind, so I'm going to kind of go through this quickly, but we specialize in both in vitro and in silico modeling. And some examples would be we do have a blood-brain barrier on a chip. We have a cornea on a chip, triculture. We've developed a lung on a chip, as well as a placental model, and also a bone and joint model. We do complement our modeling with high-fidelity physics models, including pharmacophysics and pharmacodynamics. But we have extensive experience with barrier models. And some of the key features that I'll point out later on in this are we have slits in between some of the adjacent channels that allow for cell-cell interaction, or we even have pillar designs if someone would be so inclined to have a monocyte model to explore migration or adhesion assays. So I know we've already been over what we learned with tissue chips in space, but I'll briefly kind of go over what our key findings were, and that was that you get accelerated disease progression and accelerated aging in space. And these are conditions that are difficult to replicate on Earth just because of the fact that it's in microgravity. And what we wanted to do is we wanted to explore disease, aging, and astronaut health with respect to with respect to women's health, and in particular, we'd like to study women's reproductive health or uterine aging. Each of these aspects can impact uterine health and reproductive health, and we wanted to adapt our spaceflight or adapt our model for spaceflight to assess these effects of microgravity while introducing a drug repurposing pipeline to see if we can either reverse or stop the effects of aging in the uterus. So to study this, we wanted to create something that we call Uhura, which is a microfluidic human uterus on a chip to study reproductive aging. And we wanted to adapt this model for long-term spaceflight. And that means, and in order to do that, we'd also like to recreate the menstrual cycle. So it was necessary to build upon our already established triculture model that we had of this by adding in the myometrium. And this was, it was necessary for us to reestablish basement membranes, flow rates, media compositions, cell seeding density, et cetera, et cetera. We had to kind of completely redo this thing to establish or to prepare it for long-term spaceflight. And we originally only adapted the triculture for about 7 days in culture, but we want to get this out to 35 days to completely recreate the menstrual cycle. So I wanted to, I just wanted to give a brief overview on the order in which we seed the cells and do things like increasing flow rates, et cetera, et cetera. So on day 0, we start to establish the endometrium, which is by seeding the endometrial epithelial cells into the top channel there, the EEC. And that's, we allow them to establish for about 24 hours. And then on day 1, we seed the uterine smooth muscle cells down to the very bottom channel and establish the myometrium. After the myometrium has been established, we'll go ahead and seed the epithelium, or the endothelium, with the uterine microvascular endothelial cells. And then eventually we'll seed the fibroblasts last to fully establish the endometrium. At the same time, we will begin what we call a ramp, or gradually increasing the flow rate of media fed into these channels. We'll gradually increase that flow rate over time to a constant rate. And then on day 4, we'll do the same thing. We'll do the same flow rate increase over time. Also, as a reproductive tissue, it's pretty important to incorporate physiologically relevant hormone cycles. And in doing so, we instituted a hormone dosing regimen of estradiol and progesterone. And we based this off of literature review for what the serum concentrations are, and down calculated or adapted these concentrations for use in a microfluidic model. And then we adapted these concentrations to be in sync with what the crew on the ISS would be doing. So every 5 days, we'll be having an increase or decrease in specific hormones. And the media bags will be already prepared with the hormone. All they have to do is just switch them out, starting at 10 days, 15 days, 20 days, 25 days, and so on. So finally, what we have is a fully established quad culture. So we have 4 cell types in a microfluidic chip, as you can see here on, through phase microscopy. On the top, we've got our epithelium. Adjacently, we have our stromal fibroblasts, the endothelium, and then finally on the bottom, the smooth muscle cells. And I don't know if, well, I don't know, but I'm going to point it out to you. Here in these little slits, you can see between each cell, these are about 3 to 5 micron wide slits. And this allows cells to communicate with each other or signal to each other should they need to do that, which we think is kind of an important aspect of our model. And in establishing this model, we also wanted to make sure that we had a good and viable model. And so we performed viability, such as a dead-all, using like a Newt green contrasted with hooks to check our viability. And in all 4 cell types in the quad culture, we have over 90% viability. So we like to think that that gives us a kind of a robust model. But in addition to, checking viability, we wanted to make sure that our cells were expressing the proper phenotypes. And with the proper for the proper phenotypes, we'd like to stain each cell type for certain biomarkers that are specific to those cell types. So on the epithelium, we've got EpCAM, which is an important cell adhesion molecule. In the stromal fibroblasts, we have laminin, which is a basement membrane secretion. In the endothelium, we've got CD31, which is an important junctional protein. And in the smooth muscle cells, we have alpha smooth muscle actin, which gives us our cytoskeletal structure. To further validate the model, we wanted to check the function of our tight junction. So what we did was we ran a permeability assay using fluorescent molecules like fluorescein sodium salt or 40 kilodalton tritidextran, ran those through either the epithelium or the endothelium, and measured the permeability index, which is a ratio of the fluorescent input to the fluorescent output, or how much of the fluorescence had migrated across the membrane. And this gives us an indication of how tight our junctions are and how good our tight junction formation is. So moving on from that, after establishing the model and validating, we needed to integrate this with the MVP that Redwire has developed. And so what we have to do is we have to miniaturize our model even further, which consists of our chip, which is already small, thankfully. We have a syringe pump, which is quite large and heavy, and an incubator, also quite large and heavy. Lots of tubing, CO2 gas, and we have to collaborate with Redwire to get them to integrate all of these things into something about the size of, I would say, tissue, and maybe half of a tissue box. We've got in the MVP, we'll be able to monitor oxygen, pH, and cell confluence. And in the integration, though, we do need to re-optimize the pumps. We need to optimize sensor locations, bubble mitigation. Bubbles are kind of a problem, so we don't want any bubbles in our model. Then we also have to make sure that we can operate within their parameters for media infusion and also ISS crew media collection. So speaking of monitoring cell confluence and health, we have these impedance sensors that we've used inside of on a PDMS chip. And we tried two separate experiments. These are actually well-played experiments. But we used a slower growing smooth muscle cells seated at a slightly lower confluence or seated at a slightly lower density and monitored their growth over time. And on this graph here, you can see we have impedance over time starting at 0 days where you have kind of a lag phase and then a sharp, steep growth rate. And then confluence where you can see that the graph starts to plateau. And in addition to this, we did the same thing with epithelial cells. However, we seeded them much more densely, and they grow a little bit faster than the smooth muscle cells. And this is over 24 hours. We see the exact same thing with a lag phase, a steep growth rate, and then confluence. So we wanted to also see if we could find a way to repurpose some drugs to either stop or reverse uterine aging. And we had our bioinformatics team kind of look at a public uterine data set from a GTX database. And they used a signature reversion to kind of down select or select drugs that look like they would be good candidates for reversing or perturbing the uterine aging pathway. Two of the candidates were bortezomib and palbociclib. And it turns out that these two drugs can have a reversing effect for senescent cells. They're typically used for cancer treatments, but this is kind of why we're doing this drug repurposing. So what we want to do is we want to be able to test these drugs. And what we have to do to test these drugs is we'll need to artificially senesce the cells that we are going to be studying. And currently what we're using for senescence is we're using hydrogen peroxide to induce senescence into the cells at varying concentrations. And here on the left there is this is a beta-galactosidase assay in the green, which is an indicator of senescence. And also we look at something called nuclear morphometrics. So as you age, your nuclei kind of change their morphology. And we used a pipeline from Nature paper that allowed us to assess the nuclear morphology and determine the level of senescence. And that's this chart that you have up here on the right. So in media, this is the level of senescence. The senescence level is low. And as you increase your hydrogen peroxide concentration, you trend up towards a more senescent cell based on the morphology of the nuclei. And also, we want to start eventually comparing young uterine cells versus aged uterine cells to see if we can tell a difference there within our model. And future plans are we would like to get our culture extended out to about 35 days. We need to fully characterize the aging and then start exposing in ship the drugs that we have found as potential repurposing candidates. And I'd like to give thanks to PI Carrie German, Katenbot, Kevin Ream, Dustin, and Katie for their work. University of South Florida, Matt Anderson and Begum Methik. At Tufts, Juan Yeko, he's been great giving advice on how we need to treat our cells. Shoshao at Rutgers also been critical for us. And at Redwire, Ann Rogers, Rich Bollinger, Grant Vellinger, and at Synvivo for providing the chips that we use, Gwen Fuel and Jenna Rosano. Thank you. Yes.
Are there any questions?
It's a great talk. I have a quick question on the two drugs that you trying to repurpose
Yes.
on your in vitro model. What is the dosing that you saw some efficacy? What is the concentration?
I do not have that data in front of me right now. It is I believe it is a low micromolar dosing. It was our bioinformatics team that kind of provided us with that data. But if you want specifics, get with me afterwards and I can forward
I think
that
in
to
our
you.
experience, a lot of times the dosing is just way too high compared to the Cmax that we can hit in our patients. Yeah.
Yes.
So that's why I was asking the dosing.
Okay. Yeah, thank you for the question.
Once more, yeah.
Okay, is this on? You were mentioning, I believe, tight junctions and also the potential of like studying these under microgravity, if I recall. Some of the talks blend together, so sorry for mixing up details.
No problem.
I'm particularly interested in the formation of like myomata and how like mechanical transduction might be involved in that. Do you have like the capacity to maybe comment on potential roles of mechanical transduction in uterine aging?
I don't think I have the capacity to comment on that, unfortunately. So my role is to essentially build the model to optimize the basement membrane, seating densities, cell types, things like that. As far as your question, unfortunately, if you want to get with me after this, I can take your question and forward it to the people who I think could answer that for you, but unfortunately, I'm not really good.
You mentioned ECM as well, but did
Yes.
you notice any like differential expression in ECM with any of the factors that you introduced? 'Cause
Well,
it's
what
also,
we
like,
did
fibroid.
notice was with different ECMs, we have some of the cultures would last a little bit longer in chip with different ECMs. We use this thing called an ECM array from Advanced Biomatrix to assess which ECM we thought was optimal. And I think it had about 36 different ECMs on there. You plate the cells on there for a day or two, and then you can basically assess either just with phase microscopy, or you can do some ICC staining for phenotype expression to assess which basement membrane or ECM you could use. And so that's how we kind of selected the basement membrane that we were going to use.
Okay, thanks.
Yep.
Thank you, Dylan, for this exciting presentation.